Hello everyone. The next chapter that we are covering in Physical Pharmacy 2 is Biopharmaceutics. What are we going to study in this chapter? We are mainly going to study what is biopharmaceutics and pharmacokinetics, then what is the concept of ADME, absorption, distribution, metabolism and elimination, then what are the factors that affect absorption and what are the different mechanisms of drug absorption. Okay, so these are the things we are going to cover in the coming slides. So biopharmaceutics is a major branch of pharmaceutical sciences concerned with the relationship between the physiochemical properties of a drug in dosage form and the pharmacologic, toxicologic or any clinical response observed after it is administered. In short, biopharmaceutics mainly involves drug absorption process. The systemic absorption of most drug products consists of a succession of rate processes as disintegration of a product, then dissolution of the drug in aqueous environment, absorption across cell membranes into systemic circulation, and ultimately to its site of action. Several key hurdles such as aqueous solubility, permeability, and first pass effects must be overcome so as to get a drug from a dosage form to its site of action. These hurdles now vary in how easily they can be overcome by formulation efforts. When it becomes evident that a potential drug candidate has poor biopharmaceutic properties, due consideration must be given to its developability. See, for a drug to exert its therapeutic effect, it has to undergo a lot of steps and a lot of phases. Okay, it has to be transported by body fluids. Then, if there is any kind of microbial attack or metabolic attack, it has to endure that also. Traverse required biological membrane barriers. Then it also has to avoid any kind of distribution where it is not required to escape widespread distribution to unwanted areas, penetrate in adequate concentration to the sites of action, and then interact in the fashion what is its mechanism of action. So, what happens is once when a drug is administered, its absorption starts. The drug does not remain in a single body location. It is distributed throughout the body till it is eliminated. Okay, if you can see in this diagram, once a drug has been taken in a delivery in a dosage form via oral administration, its entry into the GIT, okay, a portion of the drug is absorbed into circulatory system from where it is distributed to the entire body fluids, tissues, organs. Okay, from these sites, then the drug returns to circulatory system and then eliminated through the kidney as such, or it goes for metabolism. Where does metabolism takes place? In the liver, okay, or other cellular sites as well. Then it is eliminated from the body either directly or in the form of its metabolites. So next thing if you see in the diagram is administration of intravenous injection. Intravenous injection are introduced directly into the circulatory system, avoiding absorption, which is required for systemic effects from all other routes of administration. Various locations now to which the drug is traveling once administered, these can be understood as separate compartments. Okay, each compartment containing some fraction of the administered dose of drug. Now, the transfer of drug from blood to other body locations is generally a rapid and reversible process. Okay. Uh, by this, I mean to say, when I say reversible, I mean to say that the drug may diffuse back into the circulation. The drug in the blood therefore exists in equilibrium with the drug in other compartments. However, in this equilibrium state, the concentration of drug in the blood may be quite different than that of the concentration of drug in other compartments. So this information is going to help you understand the coming slides. So what happens when a drug is taken? The absorption, distribution, metabolism, and elimination of a drug from the body. These four processes are dynamic processes. These continue once from the time once the drug is taken till it is completely removed from the body. Okay. So the rates at which these processes occur affect the onset, intensity, and duration of drugs activity within the body. So, the area of study that elucidates the time course of drug concentration in the body and tissues is determined by is termed pharmacokinetics. Okay, so you see, because one drug may also alter the ADME of another drug, the pharmacokinetic studies are applied in the study of drug-drug interaction also. Bioavailability. According to the US FDA, bioavailability describes the rate and extent. Remember these two words, rate and extent, to which the active drug ingredient is absorbed from a drug product and becomes available at the site of drug action. Okay. Now, because pharmacologic response is generally related to the concentration of the drug at the site of drug action, the availability of a drug from a dosage form is a critical element, of a drug product's clinical efficacy. But drug concentrations usually cannot be measured as such directly from the site of action. Okay, so it is considered that the drug at the site of action is in equilibrium with the drug in the blood. Therefore, most bioavailability studies involve the determination of drug concentration in the blood or urine. When we say that the drug at the site of action is in equilibrium with the drug in the blood, we do not mean that the drug concentration in blood and tissues are equal. No, we just assume that the equilibrium is maintained and that blood concentrations are proportional to tissue and active site concentrations. Thus, bioavailability is concerned with how quickly and how much of a drug, remember the two words rate and extent, how quickly and how much of a drug appears in the blood after a specific dose is administered. That is why we say rate and extent in the definition. Now, starting with absorption. Absorption relates to the mechanism of drug input into the body and into a tissue or an organ within the body. After the drug enters the systemic circulation, it is distributed to the body's tissues. This distribution depends on many factors, including blood perfusion, cell membrane permeability, tissue binding, plasma protein binding, etc. The penetration of a drug into a tissue depends on the rate of blood flow to that tissue. Okay, it also depends on the partition characteristics between blood and tissue. When the entry of the drug and the exit of the drug, these rates become same, the distribution equilibrium between blood and tissue is reached. It is reached more rapidly in a richly vascularized area because again it depends on the blood flow. It reaches more rapidly in richly vascularized areas than in poorly perfused areas until diffusion across membrane barriers is rate limiting step. After the equilibrium is reached, bound and unbound drug concentration in these tissues and extracellular fluids are reflected by plasma concentrations. If we talk about metabolism, metabolism is the major process by which foreign substances, including drugs, are eliminated from the body. During metabolism, a drug substance may be biotransformed into a pharmacologically active or inactive metabolites or both. This is where we use the example of prodrugs. Now let's talk about elimination and excretion. Elimination, you see, relates to the chemical transformation and or physical removal of drug from the body. Hence, elimination is the sum of processes which is processes related to drug loss from the body. That is metabolism and excretion. Metabolism and excretion occur simultaneously with distribution, making the process very dynamic and complex. Excretion is the process by which a drug or metabolites is removed from the body without further chemical modification. Three primary routes of excretion occur through bile, intestine, and kidney. Bile, we call it biliary excretion, intestine and kidney we call renal excretion. I repeat, disposition can be broken down into distribution and elimination, and elimination is the sum of processes related to drug loss from the body, namely metabolism and excretion. Now let us quickly revise a few definitions. First, biopharmaceutics. Biopharmaceutics is defined as the study of factors influencing the rate and amount of drug that reaches the systemic circulation and the use of this information to optimize the therapeutic efficacy of drug products. If we talk about absorption, absorption is the movement of drug from its site of administration to systemic circulation. Bioavailability, rate and extent of drug absorption. The movement of drug between blood and extravascular tissues is drug distribution. Pharmacokinetics, it is the mathematics of the time course of absorption, distribution, metabolism and excretion of drugs in the body. Then pharmacodynamics. Pharmacodynamics and pharmacokinetics you also read as pharmacodynamics is what drug does to the body and kinetics is what body does to the drug. So we can also define pharmacodynamics as a study of the biochemical and physiological effects of drugs and their mechanism of action. Then two new terms, bioequivalence and therapeutic equivalence. Bioequivalence is talking about chemical equivalence. What is a chemical equivalent? Chemical equivalent is equivalents are the drug products that contain same compound in same amount and that meet current official standards. Okay, these are chemical equivalents. Bioequivalence refers to the chemical equivalents which, when administered to the same person in the same dosage regimen, resulting in equal concentration of drug in blood and tissues. This is bioequivalence. Whereas if we talk about therapeutic equivalence, it refers to the drug products which, when administered to the same person in the same dosage regimen, provide same therapeutic effect or toxic effect. Bioequivalent products are expected to be therapeutically equivalent.